Hey there, A&P students, and welcome to week one of our virtual lecture. This will be the format from here on out, but again, I encourage you all, if you have any questions, comments, please direct those to my Momentum email preferably. That's the one that I've been checking more often because I'm often also using Momentum to access our course websites. But if you email me through RadarNet, I can also get through to you. And if essential, I've provided you with my phone number, so you can also get in touch with me that way. Also, I will be updating a discussion board soon on Momentum with questions related to our lecture content, just so that we can have some, more specific interactions related to class content. So the featured content for today is especially relevant due to this coronavirus. So of course we're talking about the respiratory system now, which is the system that's affected by the disease COVID-19 caused by the coronavirus. But we're going to talk generally about the anatomy, the physiology, and how oxygen and carbon dioxide are transported throughout the body. So let's go ahead and start and do kind of a brief overview of the anatomy of the respiratory system. So I've had some time, a little more than we typically have in class, so hopefully you enjoyed these drawings. They may be a little bit more detailed than the ones I do in class. So what I have here is an individual, and you can see that I have drawn and labeled certain parts of the respiratory system. So let's just go ahead and think about what happens when you inspire. So that's one of the processes of ventilation, or getting air in and out of your lungs, is inspiration. And usually when we're going through the parts of the respiratory system, we'll start from where air comes in and proceed to where it ends up, where gas exchange is occurring. So air is inspired either through these nasal or oral cavities, your nose, your nostrils, or your mouth. And if you happen to inhale through your nose, that air is going to flow through the nasal cavity, through the mouth, through the oral cavity. And these are separated by the palate. You have the hard palate that is called the hard palate because it's made of bone underlying the tissue of the hard palate. And then the soft palate, which is not made of bone. But these are going to separate the oral from the nasal cavity. Something to consider about the nasal cavity in particular is that there are ridges inside the nasal cavity as well as sinuses. And these serve different purposes. So we're thinking about the structure and the function and how those relate to one another. Well, these ridges inside the nasal cavity, called meatuses, are going to make airflow through the nasal cavity turbulent. Now what that means is the air is going to be bouncing around off the walls inside that nasal cavity. And the more it bounces around, the more opportunity there is for this nasal cavity to trap dirt, debris, whatever you might be inhaling that's not meant to progress into the lower respiratory tract. So one of the big functions of the nasal cavity is to filter the air that we're breathing. So your car has a filter on it to keep the air clean in the cabin and in the engine. Your body also has a filtration system to keep the air that you're breathing clean, so that we're not introducing pathogens or debris into this vital area down lower in the respiratory tract. So turbulent airflow is good because it provides more opportunity to filter. Another thing that's important to note about the nasal cavity and this turbulent airflow is it's going to warm and moisten the air that's coming into your body. So we don't want the mucus in our respiratory tract to dry out. We want to keep it moist. And so in order to keep these, your lungs moist, the rest of your respiratory tract moist, the air that we're breathing needs to be somewhat moist, and a lot of that moisture is picked up due to this kind of turbulent flow through the nasal cavity. Also, the air flowing into your body is going to be warmed inside of the upper respiratory tract. So both the oral and the nasal cavity can warm and moisten the air that's going to be taken into your body. So once that air is passing through the nasal and the oral cavity, you come to this segment called the pharynx, which is the next site of the upper respiratory tract. And the pharynx is where the airflow is going to progress next. And the pharynx is broken into the side of the pharynx that's going to be directly connected to the nasal cavity. And on lower down, the oropharynx is going to be the part of the pharynx that's connected to the oral cavity. So you have nasopharynx superior to the inferior oropharynx. And another term for the pharynx is just the throat. So anytime anyone refers to their throat, what they're actually, the structure that they're referring to is typically, in a clinical sense, it would be the pharynx. On down, the pharynx is actually also going to be continuous with the esophagus. So that's actually where the food is going to go. Air should not be going down your esophagus. Instead, it's going to be diverted into the larynx. So the larynx is commonly referred to as the voice box. And it's called that, that's its common name, and it's called that because the voice box contains ligaments. And when air passes across those ligaments, it's going to cause specific audible sounds to be made. It's going to change the pitch of your voice. So actually making different words and things like that, that's, you see me making different words right now, that's control, muscular control over the shape of my mouth and my tongue. The pitch of your voice, however, is controlled by your larynx in the voice box. And that's kind of this a similar function of guitar strings. You tighten up a guitar string and it's got a higher pitch. You loosen it up and it's got a lower pitch. That's exactly what's happening with these ligaments inside your larynx. You tighten them to make a higher pitch, and there's muscles that pull and tighten those, or those muscles relax and loosen the ligaments within your larynx and make a lower pitch. One interesting difference, I'm sure you know that males tend to have a lower voice than females do, and that's due to the size of those ligaments. So they grow in size and length due to growth hormone differences in sex hormones. And due to that increased size and length, generally the sound that they make is going to be a lower pitch. So that's why males have a lower pitched voice. On past the larynx is actually going to be continuous with the trachea. And one thing that's important to note about both the larynx and the trachea is there's a lot of cartilage in these structures. In the trachea, you're going to see these rings of cartilage. So I've kind of drawn these little lines along the trachea. The trachea is your windpipe, and the windpipe needs to be open. It needs to be a term called patent. And patency just means how open something is. So we don't want your windpipe to close. We want to keep it patent. And to do that, we have these rings of cartilage that are going to keep the windpipe open. Wind or air that you breathe in is going to progress down past the trachea. And so another thing I'd like to mention is that now we've gone from the upper respiratory tract, which goes all the way down to the larynx. It's considered the upper respiratory tract. And the trachea and below is going to be considered the lower respiratory tract. So once air has moved into the trachea is going to divide into what's called the bronchial tree. And the bronchial tree is called that because it has many branches. The first branch is going to divide the trachea into the left and the right bronchi. And bronchi is plural for bronchus. So each of these, the left and the right bronchus. Now once that bronchus enters the lungs, it's going to further branch off into smaller and smaller branches. Eventually, those branches are going to be very, very small. And this happens, there are about 26 branches, a series of 26 branches, before you get to the smallest subset of the bronchial tree. And those, once they're at their smallest, those are called bronchioles. So bronchioles are going to be these very small branches on the bronchial tree, and they're actually what is delivering air to the site of gas exchange, the alveoli, the air sacs. So alveoli, what they look like, are going to be these little sacs filled with little, what look like bubbles. And these are going to expand as they fill up with air, and all of these all of this membrane that makes up these circles in these air sacs is a site for gas exchange with a blood vessel. So as you breathe in, oxygen and other gases enter these air sacs, and because of the pressures of oxygen, which we'll talk about in a little bit about why exactly oxygen is moving from the air into the bloodstream. But these membranes that make up these alveoli are the site of gas exchange. And of course, that is the number one function of the respiratory system, is gas exchange. So that includes taking oxygen into the bloodstream and removing carbon dioxide from the bloodstream. Another important function, and generally gas exchange, is called respiration. And respiration involves both inspiration, breathing in, and expiration, breathing out. Another, of course, I kind of alluded to it earlier when we were talking about the larynx, is speech. So speech is another really important function of the respiratory system. What separates us from the animals is our ability to communicate, not just accessorize. But communication is another really important function of the respiratory system, and it's allowed because of specific structures, largely in the larynx. And finally, if you look closely at the nose area, what you have also in the nose, one thing you know, besides bringing air into your body, it's also there to help you detect odors. So detection of odors, there are specialized neurons in the nose, and they have receptors for specific molecules and transmit those signals to the brain, which interprets as smell. So odor detection, again, another very important part of the respiratory system. So that's just a general overview, and now let's move on into some more specific features. So I'd like to go into a little bit greater detail on each of these structures, and I want to start with the nose. So once you've gone through the nares, or the nostrils, you enter the vestibule of the nose. So this little area would be called the vestibule. If you're picking your nose, you can get in there. But the vestibule contains things like hairs that are going to be there for, again, filtering debris. And there's also going to be some associated mucosa further on. So some of that might escape into the vestibule. But the vestibule is largely going to be protected on the inside by a layer of stratified squamous epithelium. So that means it's strong. It can resist mechanical forces. Whereas if you go in deeper, you get to the point where there's actually going to be what's called a mucosa. So what mucosa is, as the term kind of denotes, as you might be able to tell from the name mucosa, it's involved in mucus production and movement of mucus. So what the olfactory and respiratory mucosa is, olfactory just kind of refers to those olfactory receptors and olfactory neurons that help you to smell. But the olfactory and respiratory mucosa, what it is going to be a pseudostratified ciliated epithelium. So once you get past this vestibule and into the inner nose nasal cavity, you get this olfactory mucosa, and further down respiratory mucosa. It's pseudostratified because it kind of looks like there might be multiple layers of cell, but there's not actually. But these are going to be columnar, so they kind of look like tall columns. And this epithelium is going to include a special type of cell called a goblet cell. So I've drawn goblet right here. And it's called a goblet cell because it kind of looks like a goblet, one of those wine glasses that kings and queens drink out of. But this goblet cell produces the mucus. And because this epithelium has cilia again are those hair-like projections on the tops of these cells that's going to beat forward in one direction and move mucus in a particular direction. So within the nasal cavity, typically these cilia are going to push mucus down toward the pharynx, where you can either spit it out, or sometimes people swallow the mucus. That's also an option. Fortunately, the enzymes and acidic nature of our stomach will protect us from any pathogens that we might swallow that way. So it's okay. It might be a little gross, but it's really not a big deal, medically speaking. So these goblet cells produce the mucus. The cilia are going to propel it. So this mucus is just going to trap those debris. This is another mechanism for filtration. I mentioned the meatuses that provide that turbulent flow, and there's structures called conchae that are also involved in generating this turbulence within the nasal cavity. And there are also sinuses. So these sinuses are going to be spaces, hollow spaces, that are going to really mostly affect your ability to project your voice. They make your voice louder. They amplify your voice. So that's one reason why if you have a sinus infection, that space is filled up with mucus. You might notice that your voice is not, you're not able to project as well as you normally are if you have a sinus infection. So there are pros and cons to having that mucosa present, but we can eliminate it by adding pressure and pulling mucus in one direction or another to get rid of it. So its primary function is there to trap debris and things that might be harmful if they were to progress further down. Now I'd like to talk a little bit about the throat or the pharynx. So I mentioned before that the pharynx has kind of, what I mentioned before was the naso and the oropharynx. But there's also a third region of the pharynx, the laryngopharynx, which just opens to the larynx. So the purpose of the nasopharynx, again, is kind of the same as the purpose of the nasal cavity, is to warm the air, to moisten the air, and to trap pathogens. So in effort to do all of those things, the type of cells that you'll find here in the nasopharynx are going to be, again, those pseudostratified columnar ciliated epithelial cells, including those goblet cells. You're also going to find the pharyngeal tonsil, and a tonsil is a lymphatic organ that's going to trap pathogens. Moving on to the oropharynx, behind the oral cavity, you have a shift from that pseudostratified columnar epithelial to now a stratified squamous epithelia. And anytime you see a stratified squamous epithelium, you know it's there for protection, first and foremost. The reason for this change once you get to the oral cavity is think about what else is going through the oral cavity and traveling through the oropharynx. It's not just air, but it's also food. And food traveling through has the capacity to do damage to the cells that it comes in contact with. So because of the food that travels through this cavity, the types of cells changes in order to protect against any damage that might be done by passage of food through that cavity. Also in the oropharynx, you're going to have 3 more tonsils, which again are those lymphatic organs that are there to trap pathogens. And often because they trap pathogens and because your mouth and your nose are one of the primary areas where pathogens are able to enter your body, these tonsils can become more easily overwhelmed than maybe other parts of your immune system or other parts of the body, and are prone to infection. So sometimes people get their tonsils removed, other times you just take an antibiotic and hope the infection passes. And then again, the final part of the pharynx or the throat is going to be the laryngopharynx, which just opens up to the larynx and is going to allow air to pass into the larynx. Now, one other important structure that's going to be kind of associated here is the epiglottis. So when you swallow, the epiglottis, this little flap of elastic cartilage that is going to close off the windpipe during swallowing. And so the trachea will not be receiving air during swallowing. And sometimes if you're trying to talk and eat at the same time, this mechanism can fail, and you might have some issues there. But the epiglottis, again, is to kind of protect food particles from going down and proceeding down into the larynx. So now moving on to the larynx. The larynx, again, is going to contain the vocal cords. But the larynx is also going to contain lots and lots of cartilage. So there are several distinct bits of cartilage that we'll talk about. The thyroid cartilage is kind of the most prominent cartilage structure in the larynx. And that's going to give the appearance of what's called the Adam's apple commonly. So the Adam's apple is actually going to be mostly the thyroid cartilage, and it's called the thyroid cartilage because beneath that cartilage lies the thyroid gland. Now also in this area you're going to have the cricoid cartilage, which is going to attach to that Adam's apple by the cricothyroid ligament. The cricothyroid ligament that attaches this cricoid and thyroid cartilage is actually what will be poked through if you need a cricoidotomy, cricothyroid ligamentotomy. So if you can't breathe, they're going to poke a hole right through this ligament between the thyroid and cricoid cartilages. You also have the arytenoid cartilages and the corniculate cartilages, and those two are going to be basically their function is to aid in sound production. This is the voice box we're talking about, so sound production is important. And you also have what are called cuneiform cartilage, and these are just simply going to support this epiglottis up here that opens and closes to control whether or not air will be allowed to enter the trachea. And again, this is because during swallowing, the epiglottis is going to close off the trachea so that you don't get food down your windpipe. So I've kind of lumped these together in 2 groups, and basically the reason for that is that the thyroid, cricoid, and most of the arytenoid cartilages are actually going to be hyaline cartilage, which is the type of cartilage that usually you think of being associated with the ends of long bones that protects against friction at a joint. Whereas the corniculate and the cuneiform cartilages are going to be more fibrous, elastic fibrocartilage. So a subtle difference in the types of cartilage that make up those different structures. An important part about the larynx are what are called mucosal folds. So if you looked closely inside the larynx, you would see that it's not a smooth, the epithelia is not smooth, and that's because underlying those mucosal folds are ligaments. So there are what are called vestibular folds, and these vestibular folds are called the false vocal cords. You might assume that these folds are going to be the vocal cords, but they're false vocal cords. Beneath those you have the true vocal cords, and under those vocal cords are those ligaments that I mentioned before. So when the muscles of your larynx contract or relax, they're either going to stretch or relax those ligaments, and when air passes over those again, it makes sound and changes the pitch of your voice. So that's basically the main function of the larynx. It is a conduit through which air travels, but it's also going to be key in controlling the sound of your voice. The next feature of the respiratory tract, and now we are progressing into the lower respiratory tract, is the trachea. So past the voice box or the larynx, you have the trachea. It extends kind of from here to here. So what you'll again notice about the trachea is it's got these rings of cartilage that keep it open or patent so that air can flow freely. So these are called C-rings, and they're called that because if you were to look at a cross-section of the trachea, you would see behind the trachea is the esophagus. And so these C-rings surround the trachea, these rings of hyaline cartilage, and keep it open, but they're not complete rings. And the reason for that is because when you swallow food, your esophagus needs to expand. So if these were complete rings and the esophagus was to expand, it would push against the trachea. And in order to relieve that pressure of the esophagus expanding during swallowing of food, these rings are incomplete. That allows it to kind of close off the trachea a little bit, just so that the esophagus can expand when you're swallowing a bolus of food. A bolus is just what we call kind of a piece of food that we're swallowing. So when you swallow that food, the esophagus can expand because of these incomplete C-shaped rings of hyaline cartilage that keep the trachea open. Now if you move on down to the base of the trachea, you'll see kind of a special ring of cartilage called the carina. And the carina is going to kind of attach itself to the point. It's going to be the point where the trachea splits into the left and the right bronchus. So the carina actually also is going to contain receptors that are going to respond to certain dust, debris, allergens being trapped in this area and initiate a really violent cough reflex. So the carina is kind of the base of the trachea, again, where you're going to split into the left and right primary bronchi and further get down into the bronchial tree. The mucosa that makes up the trachea is going to be this pseudociliated columnar mucosa again. So there's going to be mucus production here. No food should be entering, so we don't have to worry about damaging that mucosa with, or those epithelial with food particles. But it is still going to be a place where mucus can trap debris. And the cilia are, instead of where you saw the nasopharynx, cilia are kind of pushing mucus in this direction toward the esophagus or out, the oral canal, the oral cavity. The cilia in this region of the trachea are going to be pushing mucus toward the throat again. So up instead of down. And that's, again, just so that mucus can either go out spitting, or swallowed, where it can be destroyed in the stomach. So now what I'd like to talk about is the bronchial tree. Now we're getting closer and closer to that respiratory zone where you're actually getting gas exchange occurring. And remember, that's happening in these air sacs, the alveoli. So first, when your tracheae breaks off into the left and the right primary bronchi. So these bronchi are going to, the trachea branches into the left and right primary bronchi. The primary bronchi are going to branch off into secondary bronchi. The secondary bronchi are going to branch off into what are called tertiary bronchi. On, and this happens about 26 times. So about 26 branches. Until you get to the smallest division of the bronchi, which are called bronchioles. So kind of like arteries divide and break until they become arterioles. The bronchi branch and get smaller and smaller until they become bronchioles. And you have about 65,000 of these per lung. So you have lots and lots of tiny little passageways, these bronchioles. So as you progress down past the carina, once you enter into the bronchi, those rings of cartilage that you saw in the trachea, there's also going to be rings of cartilage present in those bronchi. But instead of being C-shaped, these are going to be complete rings, so a complete circle as opposed to a C-shaped. And that's because there's no esophagus that needs to expand that's passing alongside these bronchi. So complete rings of cartilage are going to keep those bronchi open. And unfortunately, we won't be doing a lung dissection, but it's pretty cool to see what the bronchi look like if you cut open the lung. They're these really kind of stiff, and they're stiff for a reason, to keep them open, these passageways that air flows into the lungs. So in the bronchioles, as you progress from bronchi down to these smaller branches, these bronchioles are able to control the flow of air, how much air gets into these alveoli, or how little. And that's going to be controlled, as you might expect, based on the needs of the body, and that's often going to be related to whether or not you're in a fight or flight or a rest and digest type situation. So that's, as was the case with the other passageways for blood, it's going to be controlled by smooth muscle in the respiratory system. So smooth muscle contracting or relaxing can control the flow of air in the bronchioles to the alveoli, which again is going to be where the gas exchange is taking place. So as you progress and get smaller and smaller, the cartilage becomes less and less frequent, and also it changes in its shape a little bit. You get less of those complete rings and more kind of just patches of cartilage. And also the amount of smooth muscle is going to increase as those bronchi and bronchioles branch and get smaller and smaller. You get more and more smooth muscle because that's getting close to where the site of gas exchange is taking place. That's where you want to regulate how much air is going in and out of those spaces. So once you get down to the bronchioles themselves, they actually lack cartilage. And they also have very few cilia and goblet cells. So there's not too much mucus production going on deep in the lungs. And that's because there's a lot of filtration that's going on all the way up to this point. So most of the dust, the debris, the pathogens that you might be inhaling, those have been mostly filtered out to this point. And now we're getting to the place where we're really going to be thinking more about getting gases into and out of the bloodstream. So you have much fewer cilia and goblet cells. And finally, you get down to the alveolar ducts, where the air is actually going to go into the site of gas exchange. So the last structure that I want to talk about is the alveoli. And again, that's the site of gas exchange. That's the whole reason we're here. And so within those alveoli, about 90% of the cells that you'll find in the air sacs are going to be type 1 cells. And what those are is a simple squamous layer of epithelial or endothelial cells. And simple squamous again means they're kind of stretched really thin and they're in a single layer. So this is a very, very thin layer of cells, and that relates to its function being to allow gas exchange. So in order for gas to diffuse out of the blood and into the air in your lungs, or from the air in your lungs and into the blood, it makes sense for it to have a very thin layer of cells to cross to do that. And so having this simple squamous layer of type 1 cells achieves that goal, having a thin layer of cells. There's also what are called type 2 cells, and this makes up about 10% of the cells in your alveoli, and their only job is really to produce a chemical called a surfactant. So what a surfactant is a chemical that is going to break up the surface tension of water. So you might remember from A&P 1, we talked about how water molecules have these hydrogen bonds with one another. So water molecules tend to want to attach to one another with these hydrogen bonds. And in doing so, they can actually pull structures that they're attached to. So the surface tension of the water in your alveoli, without the presence of this surfactant, that surface tension could actually pull your alveoli closed. So if you had this normal surface tension that you see with water, you would have much less capacity to pull air into those air sacs where that really important gas exchange is occurring. So with surfactant, you break up that surface tension and relieve that surface tension, which allows them to fully inflate. So you need surfactant to fully inflate your alveoli so that you get a proper amount of gas exchange going on. And another important structure-function relationship is these air sacs are going to make up a whole lot of the proportion of your lungs. And if you were to add up, again, looking at the structure of alveoli, it looks kind of like a bunch of grapes. Each of these represents an alveolus, and the membrane here is where gas exchange is occurring. And if you add up the area of all of the membrane from all of the alveoli in your lungs, you get about 1,000 square feet, which is about the surface area of a small apartment. Actually, that sounds like a pretty big apartment to me. Normal-sized apartment, we'll say. So there's enough surface area in your lungs for gas exchange to occur to cover a small apartment. That's a pretty impressive number. So the structure-function relationship being you have a lot of surface area, and that makes gas exchange incredibly efficient. I also forgot to mention macrophages are present in the alveoli, and a macrophage is just a white blood cell whose job is to that they are phagocytes. They go through phagocytosis, so they're crawling around looking for debris and pathogens to ingest, to destroy. So these are some immune cells, and protecting the alveoli from infection is really important. So you have macrophages there that are going to be kind of cleaning up the debris. And once they've done that, they're actually going to travel back up the trachea, get stuck in some mucus, and take with them the debris that they've trapped. So they kind of sacrifice themselves to clean out your lungs and the alveoli. So now I'd like to spend some time specifically talking about the lungs. And the lungs are going to be located in the thoracic cavity. So the thoracic cavity is going to be bounded by your rib cage and your diaphragm. And in between the lungs, you'll have the mediastinum, where you have several other organs, including the heart, which makes it so that there's this notch on the left lung. So within the thoracic cavity, what's actually going to provide movement, or the pressure that's going to cause air to flow in and out of the lungs, is expansion and contraction of the thoracic cavity. And how we achieve this expansion and contraction is primarily going to be the movement of the diaphragm. That's also going to be assisted by the external intercostal muscles. And intercostal, again, just means between the ribs. So the muscle on the exterior between the ribs is going to be providing about 25% of the effort that it takes to expand or contract the lungs, whereas the diaphragm is doing about 75% of the work. The diaphragm normally has this curved shape when it's relaxed, and as it contracts, you get contraction of your diaphragm, it's going to flatten out, and that's going to pull the lungs down and expand the volume of the thoracic cavity. Now, one important thing to think about and what causes air to go in and out of the lungs, it's all about pressure gradients. And there's a couple of important laws. One important one is called Boyle's law. And Boyle's law states that if you have a constant amount of gas, so let's say there's some amount of gas that's in the lungs, if you increase the volume while keeping the amount of gas constant, you have the same amount of gas, but now it's in a larger container. What that's going to do is decrease the pressure. And what causes movement of gas is differences in pressure, a pressure gradient. So what's going to cause air to flow into the lungs is that the pressure inside the lungs themselves is going to be less than the atmospheric pressure, the air that we breathe in, the air around us. So only when the pressure in the lungs dips below the pressure outside from the air that we're breathing in, that's when you're going to get air moving into the lungs. Now, the opposite is also true. So this first one that I mentioned, if you're breathing in, you're actually going to be increasing, again, the volume of your lungs. That decreases the pressure inside the lungs. Now you have pressure, air moving from high pressure outside to low pressure inside the lungs. Then the opposite is also true. When you decrease the volume of the lungs, you increase the pressure. So when this diaphragm relaxes, when the external intercostal muscles relax, they're going to make the volume of the thoracic cavity smaller. And that's going to increase that pressure up above the atmospheric pressure. And so that air is then going to travel out of the lungs and out of your body. So far I've mentioned the atmospheric pressure, also the intrapulmonary pressure inside the lungs, and the gradient between those is what's going to cause airflow between the lungs and the outside environment. But there's one more other important pressure, aside from the pressure in your lungs and the pressure in the outside environment. There's inside of what's called the pleural cavity. So the pleural cavity is going to be formed by, again, a serous membrane. So remember, lots of internal organs are covered by serous membranes, which have a parietal or outside layer and an inner layer that touches the organ itself, the visceral layer. So here in green, I've drawn these pleural cavities that surround each of the lungs. And those pleural cavities, the parietal layer, or the outer layer, is going to stick to the wall of the thoracic cavity. It's going to be attached through fibrous connective tissue. And the visceral layer is going to be attached to the lung itself. And in between, you have that pleural fluid or serous fluid. Now, one really important thing is that within those pleural cavities, there is negative pressure. And without thinking too hard about it, what negative pressure means inside that pleural cavity is it's basically going to cause the two membranes of the pleural cavity to stick to one another. It's going to create a vacuum. So one part of what creates that negative pressure is that fluid is being circulated into the lymphatic system. That parietal fluid is going to circulate out. And as it's pulled out, it's going to create a vacuum. So you have negative pressure here. And so these two membranes that make up the pleural cavity are going to basically be stuck to one another. And since both of those membranes, they're either stuck to the wall of the body, the wall of the thoracic cavity, or one is stuck to the lung itself, that means when you expand this cavity, it's also going to pull the lung with it. So you expand the volume of the thorax, you're going to be expanding the volume of the lungs. Now, if you were to take a knife and jab through somebody's lung, you probably have heard of a collapsed lung before. So the lungs are very elastic and they tend to want to collapse, but what keeps them from collapsing is the fact that these pleural membranes are stuck to the body wall, they're stuck to the lung, and they're stuck to one another. So that keeps everything stretched out. If you are to rupture that pleural cavity and relieve that suction, that negative pressure, then you're going to get collapse of the lung. So that's what happens when you have a punctured, say a stab wound. Why the lung collapses is that the pressure in the thoracic cavity is then going to be higher than the pressure in the lungs, and that's going to cause the lungs to collapse. So you need this pleural cavity to be able to keep the lung from collapsing. So now what I'd like to do is talk about this graph that you see in your book. And this graph is called a spirogram. So what spirography is when a clinician of some sort probably is going to be measuring the various lung capacities that you have and looking at how the volume of your lung changes as you perform certain breathing exercises. So let's think about the normal breathing pattern that you usually take when you're at rest. So let's say this balloon is your lung. So your lung is never going to completely deflate. There's always going to be some reserve volume unless there's something unspeakable that happens. But so when you take a breath out, you're going to have some volume left. But so let's say you take a normal breath in just at rest. So let's say that's a normal breath in. You take a normal breath out. So this would be called the tidal volume, the difference in those two volumes of your lung. So as you breathe in, you breathe out, the volume in your lung changes. And that tidal volume is going to be about 500 milliliters per breath in, breath out cycle. But the normal breath that you take during a resting period is not usually as deep as it's possible for you to breathe in. There's some extra what's called inspiratory reserve volume. So let's go back to that normal tidal volume again. Normal breath in. And let's say you keep breathing in as much as you can. At some point you're going to reach a maximum. And so the difference between the normal inspiration volume and the inspiration volume for a maximum inspiration is called that inspiratory reserve volume. That's the reserve volume in your lung that's left for a maximum inspiration. So let's breathe back out now after that maximum inspiration. Back to our normal tidal volume after a breath out. So usually when you're breathing in and out, you don't breathe in as much as you can, but you also don't breathe out as much as is possible. So when you're, if they're testing to see if, if you're an asthmatic, they'll have you breathe as much in as you can, as much out as you can. So not only is there an inspiratory reserve volume, there's also an expiratory reserve volume. So let's breathe out as much as we can. Blow, blow, blow. And you'll reach some minimum volume that you can get your lungs down to without puncturing them. So the difference between your normal breath out, the volume there, and the maximum breath out is your expiratory reserve volume. That's the reserve volume that's there for expiration. And the difference between the bottom, the most you can push out, so what's the volume there, and 0, the difference between those two numbers is going to be the reserve volume. That's how much air is going to be left in your lungs after a maximum breath out. Now, if you think about the maximum breath out, the volume there, and subtract that from the maximum breath in, that is your vital capacity. That is going to be VC here. That's going to be basically the largest amount of air that you can get in and out in one breathing cycle. Now, if you go from a normal exhalation up to your maximum inhalation, and that's what's going to be happening usually during exercise. You usually don't breathe all the way out, but you're going to breathe much more deeply in. So you're going to be going, and that's the inspiration capacity, is from that normal breath out to a maximal breath in. That's something that you would see during, something like a normal exercise. And then your total lung capacity is going to be from 0 up to what your maximal inspiration volume is going to be. So that would be your total lung capacity, or TLC. So now I think it's important to talk a little bit, now that we know kind of some about the anatomy and physiology of the lungs and the respiratory system at large, I want to talk about one of the major aspects of why we breathe at all, which is the gas exchange portion, and getting at the physics of gas exchange. Why is it that gas moves into the blood, oxygen? Why does it go into the blood at all? It's related to what the pressure of oxygen is in the atmosphere versus in somewhere like the blood. So to think about why a gas moves in a particular direction, you have this Dalton's law of partial pressures first. And what the pressures that it's talking about is what is the pressure that these gases that are in the atmosphere are putting onto the walls of a container. So the pressures in the atmosphere are basically the sum of the pressures of all the different gases in that atmosphere. So in our atmosphere, it's mostly nitrogen. You have some oxygen, some argon, and other, some carbon dioxide, things like that. But the main one that we're going to be focused on would be oxygen, of course, and carbon dioxide. So the pressure of nitrogen added to the pressure of oxygen and the pressure of all the other gases is going to equal the total pressure in the atmosphere. And again, gases are going to move in a particular direction when there is a pressure gradient. So when you're talking about whether or not oxygen is going to enter the bloodstream, you would be thinking about what the partial pressure of oxygen is like in the atmosphere, in the air that you breathe into your lungs, versus what the partial pressure of oxygen is in the blood. So for oxygen to move into the blood, the partial pressure of oxygen has to be lower in the blood than it is in the atmosphere. Now, if you think about whether or not a gas will dissolve, it's really about two things, and we have Henry's law to describe whether or not a gas will move into a liquid. And the two factors that determine this are going to be those pressures that I was talking about. What are the partial pressures of that particular gas in the atmosphere versus in the liquid? And then what is the solubility? How easily does that substance dissolve? So a substance will be much less likely to move into a liquid if it doesn't dissolve as easily. And the more soluble a substance is, the more likely it is that it's going to move into that liquid. So the first type of gas exchange that we're going to be talking about would be the pulmonary gas exchange, the exchange between gases in the air that you breathe into your lungs and the blood vessels in your lungs, the pulmonary capillaries that contain red blood cells. So what you have is this, again, this thin, this simple squamous epithelium inside the alveolus. And it's so thin that gases can just diffuse right across that layer of cells. And these gases, of course, include oxygen diffusing into the capillary and binding to hemoglobin in a red blood cell, and carbon dioxide being carried to the lungs in the red blood cell and being diffusing out into the alveolus. So oxygen and carbon dioxide going in opposite directions. Now this exchange can be affected by a few different properties, and one of those is going to be, what is the total surface area? So remember, all of these membranes in the alveoli combined is about 1,000 feet squared. That's a lot of membrane. But one way to get rid of that membrane is to smoke cigarettes or do other ways that you can do damage to your lungs. So damaging those alveoli with chemicals like those that you find in cigarettes, smoke inhalation, can destroy these membranes. Can also thicken the walls. Scar tissue in these membranes can thicken those membranes, make them thicker, and make it harder for oxygen and CO2 to diffuse across those. So adding scar tissue in your lungs can also make it more difficult for pulmonary gas exchange to occur. Ventilation perfusion matching is another kind of complicated sounding term. Ventilation perfusion. So ventilation is just how much you're breathing, and perfusion is how much blood flow there is. And so your lungs and your blood vessels in your lungs are going to work together to match up how much you're breathing with how much blood flow there is in a particular region. So the smooth muscle in your bronchioles will relax to allow more air to get to a place where there's high blood flow, where there's relaxed smooth muscle in the capillary, for example, or before the capillary. So your lungs and these capillaries, the alveoli and the capillaries, or the bronchioles and the capillaries, are going to match the rate of ventilation with the amount of perfusion. And that's because they're going to kind of optimize this gas exchange. So the more blood flow there is, the more air there should be to optimize the amount of gas exchange that's going on, to make it so that your breathing can optimally provide things like oxygen and get rid of carbon dioxide. So emphysema is actually going to be, a result of prolonged smoking, where you have maybe thickening and destruction of some of these membranes, and that can again greatly reduce the amount of pulmonary gas exchange and can lead to a host of issues. So now that these gases are in the blood, where are they going? And of course that is the tissue that needs oxygen, and it needs to relieve itself of carbon dioxide that builds up during metabolism, generating ATP. So you have these capillaries, these capillary beds that are delivering basically erythrocytes carrying oxygen to a tissue that needs it. And the partial pressure of oxygen in the tissue is going to be lower than the partial pressure of oxygen in the blood. That allows oxygen to diffuse into that tissue. Likewise, carbon dioxide is going to diffuse out of that tissue and is going to be carried away by the red blood cell. So this occurs in a very similar manner to what you saw in the pulmonary gas exchange. This time it's just that the directionality is a bit different, and instead of the alveoli, you have interaction between the red blood cells in a capillary and a tissue. So I want to talk a bit more also about the factors that affect gas exchange at the level of the tissue. And those are going to be, like the surface area of alveoli had a great effect on the amount of gas exchange going on. The surface area within the capillaries is also going to be having a great effect on how much gas exchange there is. So if you look at the structure of a capillary bed, it looks kind of like a spider web. There's lots and lots of surface area for that exchange of gases, and that increase in surface area increases the amount of exchange between those capillaries and the tissue. Another effect or variable that can affect how much gas exchange to the tissue there's going to be is how much blood is flowing. So the more tissue perfusion, or the more blood flow that's supplying the tissues there is, the more gas exchange there's going to be. And finally, the distance of diffusion. So how thick is this layer of cells? Just like the thickness of the alveoli determines diffusion. So these capillaries are very thin, as we talked about when we were looking at the circulatory system. So very few layers of cells for these gases to diffuse across. Actually just a single layer of cells. So the thickness, and again, scar tissue can decrease the amount of gas exchange that would be going on there. So doing damage to these blood vessels is going to be harmful to the tissue potentially. One thing that I want to talk about specifically, so we know that hemoglobin binds oxygen, but hemoglobin is also going to be able to bind carbon dioxide and take it away. So carbon dioxide traveling in the blood has a few different ways that it will travel. And bound to hemoglobin is about 20%. So I'll shorthand for hemoglobin is HB. About 20% of the carbon dioxide dissolved in your blood is going to be bound to hemoglobin. Most of it is going to be in the form of these bicarbonate ions. So that's about 70% of the carbon dioxide that's transported in your blood is actually transported as this substance that is called bicarbonate. And we'll talk about that more in just a second. And then the remainder is going to be about 10% that's simply dissolved in the plasma. So carbon dioxide doesn't have to be bound to something. It doesn't have to be transferred or converted into bicarbonate. It can simply be dissolved in the plasma, but that's the minority. And the majority, again, is going to be this bicarbonate ion, which acts as a buffer, and we'll talk about that in just a second. So one useful aspect of having carbon dioxide around in the body is that it can be converted into what's called bicarbonate. So firstly, if you have carbon dioxide being produced, this is in the mitochondria, of course, that are making ATP, giving off lots of carbon dioxide as a byproduct of glucose metabolism. So carbon dioxide will be given off and absorbed by the erythrocytes, the red blood cells. And within those red blood cells, you have this enzyme called carbonic anhydrase that's going to basically convert CO2 in addition to water molecule into a molecule called carbonic acid. Now, this is called an acid because this reaction is favored. An acid is likely to give up a hydrogen ion. And remember, hydrogen ion concentration is what determines pH. So the more carbon dioxide you have in your blood, the more likely you are to have more acid in your blood. But we have buffers in our blood that can help compensate for this acid. But so these bicarbonate ions are ultimately formed by red blood cells and released into the plasma. And so this is how carbon dioxide is generally transported in the blood, is not as free carbon dioxide, but as this bicarbonate ion. So this HCO3 is bicarbonate. And back near, when this blood circulates back to the lungs, bicarbonate is going to be converted back into carbon dioxide when it can then be released into the air inside the lungs so that we can expel that. But so bicarbonate is generally the form that carbon dioxide is going to take. So these reactions are all reversible. They can travel in either direction. And one interesting thing about bicarbonate's ability to buffer, to protect against changes in pH, is that bicarbonate can absorb free hydrogen ions. So normally when this bicarbonate is formed in a red blood cell, that red blood cell is returning to the lungs, so it's got some free space on hemoglobin. And these hydrogen ions are actually going to be bound by hemoglobin. So they're not really contributing to a change in pH just as a result of getting rid of carbon dioxide. Now, these bicarbonate ions that have been released from the red blood cells into the plasma, they can then absorb any other free hydrogen ions that there are. So if you have an increase in hydrogen ion concentration, it's going to decrease pH. Those bicarbonate ions that you have in your blood from this carbon dioxide are going to absorb any free hydrogen ions and buffer against changes in pH. So having bicarbonate around is really useful, but ultimately this gets converted back into carbon dioxide before that blood enters the pulmonary capillaries. So this is the bicarbonate buffer system, and this is the way that most carbon dioxide in the blood is this is the form that it will take, is in this bicarbonate ion. So if you're looking inside of a red blood cell, and you see these molecules of hemoglobin, there's going to be 4 subunits, and each of those subunits of hemoglobin can bind a molecule. They can bind oxygen, they can bind carbon dioxide, they can bind hydrogen ions. So red blood cells are really good at carrying gases in the blood. And whether or not those gases will be bound to these hemoglobin, these heme groups on hemoglobin, depends on what the partial pressure of that gas is, whether it's going to stick to hemoglobin or not. So if the pressure of oxygen is really high, let's say you're a patient in the hospital who's having trouble breathing, they might feed you pure 100% oxygen to increase the partial pressure of oxygen in the atmosphere. And what that's going to do is increase the amount of oxygen that's going to diffuse into your blood, the amount of oxygen that's going to ultimately stick to hemoglobin. So if there's a really high partial pressure of oxygen, you might have an O2 bound to each of these subunits. And here we would say that this hemoglobin molecule has been saturated. So I'd like you to take a look at the dissociation curve for hemoglobin and try and make sense of that yourself. That'll be kind of a homework activity. But it's all about how the pressure of a gas like oxygen, the partial pressure of that gas, is going to affect the saturation how much oxygen is bound to molecules of hemoglobin. So typically in the blood, the partial pressure of oxygen in the arterial circulation is going to be about 75% saturated. Meaning for the average hemoglobin molecule that's coming from the heart going out to the tissues, on average they're going to have about 3 molecules, so it's going to be 75% saturated with oxygen. Now in the venous end, that's on average each molecule of hemoglobin is going to drop off 2 oxygen. So when it's returning, it's lost 2/3 of its oxygen, and you have a 25% saturated hemoglobin molecule on average in the venous system. And that's because the partial pressure of oxygen in the tissue is lower than the partial pressure of oxygen in the blood, and so oxygen is going to diffuse out to that tissue. Then you move back into the pulmonary, into the lungs, and the partial pressure of oxygen here is now lower than it is in the air that you're breathing, and you pick up oxygen again. And the whole cycle repeats itself. So the last thing that I would like to talk about is the way that ventilation is controlled, breathing in and breathing out, and how fast, what is the rate, and what is the depth of your breathing. And that's what ventilation is. So first, there are voluntary controls that will allow you to consciously control the rate and depth of your breath. And this ability lies in your cerebral cortex. So this is the decision-making, the conscious decision-making center of your brain, where you use your own voluntary controls to control the rate and depth of your breathing. And what you're doing when you use voluntary control of your breathing is you are overriding all of the other autonomic controls, the subconscious controls. So of those subconscious controls, there are 3 that we'll talk about. First is in the medulla oblongata. So the control center for many biological variables is the brain stem, and in particular the medulla oblongata has a large amount of control over some of these biological processes. But what's happening here with the central chemoreceptors? So chemo means there's some chemical that's being measured by cells in the medulla. And the chemicals that we're talking about here are carbon dioxide. So the partial pressure of carbon dioxide is being measured by the medulla, and the concentration of hydrogen ions. Basically, the medulla oblongata is measuring pH. And it's able to either increase or decrease the rate and depth of ventilation subconsciously to compensate for too high levels of carbon dioxide and hydrogen ions or too low. So if these levels were too high, you would want to breathe more forcefully and more quickly in order to expel more carbon dioxide, and vice versa. If these levels were too low, we would assume that you're not breathing enough, or excuse me, that there's not enough coming in. So the second type of neural control over the ventilation is going to be peripheral chemoreceptors. So central chemoreceptors lie in the central nervous system. These peripheral chemoreceptors are going to be found on these elastic arteries, the aorta and the carotid artery. And what they're going to be measuring is the partial pressure of oxygen. So if this dips too low, these chemoreceptors can cause a negative feedback loop that will increase the rate and depth of ventilation to compensate for that. And finally, there are pulmonary stretch receptors that can detect how much stretch there is in the trachea and the bronchi. And what their goal is to inhibit inspiratory muscles so that overinflation does not occur. So when you're inspiring, when you're breathing in, your lungs are going to stretch. And to prevent that stretch from actually damaging the tissue of your lungs, you have these stretch receptors that can inhibit the muscles that are responsible for opening up the lungs, so that doesn't harm the lungs themselves. So that does it for our overview or our crash course on respiration. I'll be posting a discussion question soon, and I'm also going to be adding a feature in a future lecture that will talk specifically about the effects of the COVID-19 disease that you'll see manifest itself in respiratory conditions.